It's my pleasure now to introduce you Dr. Susan George, who is a professor of medicine and pharmacology and toxicology at the University of Toronto and a clinician scientist in the Division of Endocrinology at the Toronto General Hospital at the University Health Network. Dr. George is a pioneer in the study of G protein-coupled receptors. Her innovative and leading-edge research included the discovery and the studies of GPCR oligomerization and the genes encoded in many GPCRs. This research is highly significant of neuropsychiatric and neurodegenerative disorders of the brain, elucidating the pathophysiology mechanisms and the potential treatments, strategies for drug and other addictions, depression, schizophrenia, Alzheimer's disease, and Parkinson's disease. She has been a member, named the member of the Order of Canada. Congratulations, Dr. George, is well-deserved. One of the country's most prestigious honors. It's a wonderful pleasure to have you here. The floor is ours.
Thank you very much, Anna. I'm delighted to be here and to start off our first symposium of the day entitled Introduction to Brain and Mitogenetics. Our plenary speaker is Dr. Martin Picard, who is currently Associate Professor of Behavioral Medicine in the Departments of Psychiatry and Neurology at Columbia University Irving Medical Center. Dr. Picard actually has an important Canadian connection. He trained in neuroimmunology and mitochondrial biology of aging at McGill University, then moved to the University of Pennsylvania for his postdoctoral training with Drs. Doug Wallace, and also worked with heavyweight scientist Dr. Bruce McEwen at Rockefeller University. He joined the faculty of Columbia University in 2015, where he directs the mitochondrial psychobiology laboratory. Dr. Picard's research is at the frontier of the brain-body communication with a focus on mitochondria. He uses a variety of investigative tools, including clinical, cellular, and computational approaches to examine how psychosocial exposures impact mitochondrial structure and function, and in turn, how perturbations within mitochondria influence key brain-body processes that sustain human health, including, very important for neuropsychiatric disorders, stress resilience, cognition, and the changes during aging. Does develop an important mitochondrial health index, identified novel membrane structures for mitochondrial communication, and showed that cell-free mitochondrial DNA is a psychological stress-inducible molecule actually detected in the blood and saliva. He also has a cellular lifespan model that recapitulates molecular longitudinal trajectories. We are delighted to hear him and look forward to his talk on energy, mitochondria, and the brain-body connection. Dr. Picard.
Thank you so much, Susan, for the nice introduction, and Anna for organizing such a wonderful symposium. Beautiful program you have. And my role here, I've been asked to tell you about some of our work on mitochondria in the brain specifically. And I think the nice little video that you saw highlights the importance of energy in the brain and in mental health. And fundamentally, I wanted to start with this general picture here. Energy is essential to fuel life and to fuel the brain, right? If we think about fundamental difference between a cadaver, a dead body, versus a thinking, feeling, conscious organism, like we have here on the symposium today, is really the flow of energy, right? The genes are the same between the dead body and the living body. By sequencing the DNA from the cadaver and the living person, you cannot actually tell who was alive or who was dead. So the life-giving substrate here is energy. And ultimately, every life forms on the planet are fueled by energy coming from the sun. And what plants do is that they use that energy beaming down as photons to transform CO2 into oxygen and then food substrates. that we breathe in and that we consume. And then this is where the magic happens. In the mitochondria, the food substrates, the energy that's kind of held between the carbon-oxygen bonds or released, the electrons are flown through the electron transport chain or the respiratory chain, and then oxygen is used as a terminal electron acceptor in the mitochondria. So this process of electron transport then provides the energy or extracts energy necessary to build a membrane potential. And this is really the charging your batteries. This is the life-giving process that happens inside the trillions of mitochondria in your cells. And in turn, interestingly, when we look at what mitochondria release and as byproducts of this energy transformation process, they release CO2, carbon dioxide from the Krebs cycle, and water, which is what happens when oxygen at the end receives the electron. Interestingly, you'll notice CO2 and water is exactly what you need to give your plants. They take the CO2 from the air, you water them, and voila, this is the cycle of life. So mitochondria are that essential to mammalian life as the photosynthetic reactions that happen in plants. And so why do we need energy? And quite simply, when we think about stress, when we think about the brain, energy is that interface where stressors from the outside world, right? The events, the exposures that we receive, which can be of various types, can be auditory, visual, metabolic. Stressors can be real or imagined. Humans are really good at making up potent stressors, can be emotional, pathogen, physical injury. Those stressors can only affect us, affect the human body, and trigger changes in the brain because the brain is energized, because the body is alive. And in turn, those stressors interact with information that's held within the organism in the form of circulating hormones, DNA, the epigenome, existing proteins, and all sorts of other things that hold memories of past exposures. So there's kind of hardware stuff that's encoded in our cells. And then there's this outside influence that come together. And the place where they come together is where energy is. If the system is dead, then stressors have no effect and the information is just inert information. But when the system is alive and moved by energy, then that can lead to stress adaptation, resilience, and beautiful changes that we call learning and memory, for example. And one important recognition is that the process of adapting and undergoing dynamic changes in the brain requires energy, and every little bit of life requires energy, and this makes it clear why mitochondria are important. So we wanted to ask a question about what's the role of mitochondria beyond energy production? What's the role of mitochondria in shaping stress response signatures? And the motivation for this is that different individuals out there in the world, right here, people attending the symposium, we all have unique ways of responding to challenges, right? The same stressor is going to cause me a certain physiological, psychobiological response that is going to be, slightly unique and different than, yours and so on. What shapes those differences? What's at the origin of this? It's pretty clear that it's not just about genes, not just about genetics, because you can have genetically identical mice in the same litter that will have different stress responses, that will have different levels of anxiety, as we'll talk about later today. So here in this study, we took mice with the same nuclear genome, but with different types of mitochondria. And I won't go into the details, but this ND6, the CO1 mice, these two mice have defects in mitochondrial energy transformation, the electron transport chain. The ANT1, the blue mice, have impairment in ATP, ADP transfer. So mitochondria can make ATP just fine, but they can't really release it as easily. And the NNT, the red mice, have intramitochondrial oxidative stress. So they cannot regenerate the antioxidant defenses in the mitochondrial matrix. So we ask, what happens if we stress mice with different mitochondria? Are they going to have different physiological responses? This is one example of a sample result where on the x-axis we have time, and then on the y-axis we have the abundance, the concentration of an important hormone in the blood of these mice. And you can see what happened there is we stress them for 30 minutes. And one way to stress mice is you put them into a little falcon tube with a little hole so they can breathe. But when they realize they're stuck, then they mount this neuroendocrine response where certain hormones go up, the heart rate increases, which is very similar to the neuroendocrine response in humans when we're exposed to what's called social evaluative stress, right? When you feel like other people are judging you, like giving a talk in front of an audience, for example, triggers this kind of response. So this is a model, this restrained stress mouse model has been used in the field for decades to study this kind of response. What we see here in black are the wild type mice with normal mitochondria. You see this beautiful increase in corticosterone over the 30-minute stress period, and then the stress ends and you see the nice recovery. Now, the blue line is what happens if you have mice with mitochondria that cannot exchange energy properly. The same stressor, but here, this HPA axis response, the hypothalamic pituitary adrenal axis that leads to the release of corticosterone, is twice as high as in control mice with normal mitochondria. And then if you cause intramitochondrial oxidative stress, this response is blunted by half. So these are large scale, large effect size modulation of critical stress response pathways just by tuning the mitochondria, and not by changing how much ATP they can produce, but by tuning other parameters of their biology. So here, this is one example of something you can profile longitudinally in mice. So we've profiled several other things, including plasma metabolomics and other hormones, cytokines, and so on. So there's 74 parameters total. Each row in this heat map is a parameter. Each column is a different mouse strain with different mitochondria. What you can see just at a glance, red means more active or hyperreactive. Light green is hyporeactive relative to other groups. So each mouse, each strain has a different unique cohort, set of activation or deactivation patterns. And then if you combine all of this information and make it more understandable in using a dimension reduction approach, for example, then what you find is that first, this model here, you have the two principal components that are reducing 74 variables into just 2 variables, so we can actually understand what this is showing. So compared to mice with wild type normal mitochondria, if you perturb mitochondria in different ways, it shifts the stress response, right? This is showing us in this 2-dimensional space how the organism of these little creatures chooses to respond to that induced psychological stress. So this study was quite impactful for me and my thinking around the role of mitochondria in regulating physiology and potentially influencing inter-individual differences in responses. And we're now in the process of doing the human version of this study, where we recruit participants with genetic defect in their mitochondria. They have a rare mitochondrial disease. We expose them to a challenge, and then we profile their stress physiology in the same way. But today we'll focus on the brain, and what's leading us to the brain is, a body of literature that we reviewed a few years ago with Bruce. And the take-home message from this review, no need to go through this table, but I just wanted to show this to illustrate. There's about 28 studies that illustrate quite well that mental stress in mice and rats affect brain mitochondria. So mitochondria have the ability to respond to those stressors, and that I was mentioning earlier. This is a little bit of work we've done in the mouse brain. Here we're looking at mitochondria. So we're flying through now a granule neuron in the mouse hippocampus. All of these colored structures are mitochondria. Look at these, how beautiful they are, how diverse they are. They're mitochondria that are little punctate, little beans. Like the textbook shows you, most mitochondria are not like beans. They're actually long tubular structures. They touch each other, they intermingle. Now we're flying out of the cell. The nucleus will reappear. And then you see this mitochondrial network really that surrounds the cell nucleus, where genes are, where the epigenome is, right? So mitochondria are positioned just around the nucleus, topologically perfectly situated to regulate gene expression and other cellular factors. So the point here is that mitochondria are not all created equal. There are different types of mitochondria in different cell types. And even within the same cell, what this paper shows that I'm not going to go into is that the mitochondria in the axons versus the mitochondria in the dendrites versus the mitochondria in the cell body are different. They actually look different, and their function is also different. So let's talk about the brain. And there are 3 main questions I'd like to address today. The first is, how do mitochondria in different brain regions functionally respond to stressors? Right? So we can use here mice that have normal mitochondria, expose them to a challenge, and then ask how do mitochondria recalibrate in response to this. Second question that we'll look into is, does mitochondrial respiratory chain function or content predict animal-human behavior? So there are differences in anxiety and behaviors between genetically identical animals. Could this be driven by differences in their mitochondria? And the third question is, can we begin then to create a functional cartography of mitochondria across the brain, the mouse brain and the human brain? So this is a little schematic, simplified version of what's kind of under the hood in the mitochondria. So you zoom in either into a white blood cell or to a brain cell. And then there's this respiratory chain that I mentioned, where the food substrates electrons are ripped off the food substrates, transported to the respiratory chain. Oxygen is there to accept it. You build the membrane potential, then you can use that to make ATP. So there's a few components here that you can measure enzymatically through, from brain homogenates. In this case here, we took complex 2 and complex 4 of the electron transport chain. There are good biochemical assays that we were able to miniaturize into 96-well plates, so you can do hundreds of samples. And then citrate synthase is an enzyme that's used widely to as a surrogate for mitochondrial content. And then mitochondrial DNA, which also is a poorer but surrogate for mitochondrial content. So then if you put these two enzyme activity that produce energy at the numerator, and then you divide this by how many mitochondria there are, it gives you this mitochondrial health index, or this MHI. That is an index of how much energy can mitochondria produce on a per mitochondrion basis. And we first developed this index to address questions related to the mind-mitochondria connection in humans. And here we studied 91 women and found that mood predicts positive mood. If people report feeling inspired and feeling love, closeness, and trust with other people around them, that predicts 10 to 15% better mitochondrial health index, better energy production capacity in the white blood cells the day after. So within 24 hours, there is this connection between subjective experiences and mitochondria. And this was evidence of a directional connection between the mood and the mitochondria. And so there's a lot more work that needs to be done in this space, but this is just one example of how this kind of tool can be deployed in accessible tissues like blood cells, and then to use to ask questions about the brain-body processes and the mind-mitochondria connection. So the first and the main part that I'll talk to you about today is a study that we did in collaboration with brain experts. My background is not, I don't know much about the brain, unfortunately, but I know some about mitochondria. So we partnered with brain experts. Chris Aneker is a mouse behavioral scientist, as well as Danny Dimitriou. Manish is a computational expert who specializes in analyzing brain-wide neuroimaging data. So we use some of his methods, computational methods, to examine some of the data I'll show you. And then we look at stress-induced mitochondrial recalibrations across brain regions and brain circuits. So here's the basic design. You take normal healthy mice, and then you expose them to two different types of stress. One is called social defeat, which means that for 10 days, you put the small black, C57 black 6 mouse in the presence of a big aggressive CD1 mouse. And then the small mouse basically gets bullied for several minutes. And then you take you separate them so that it doesn't get injured. Then you put a plastic divider with a few holes so the small mouse can still see the big mouse. And then it's under chronic psychological stress for a day. Then after a day, you put the little black mouse in with the new aggressor. And then there's this chronic stress for 10 days. So that's a widely used model of chronic stress in the mouse literature. And the other model is corticosterone. So this stress hormone that I mentioned earlier that increases with psychological stress, you can put that hormone into drinking water. Here you have the little bottle with the green water with the corticosterone. Then you can have mice drink this. So every time they drink water, they drink the stress hormone. So then they're under this constant, single simplified stressor. And then what you can do is pick several brain regions. So Ayelet, the student in the lab, was courageous enough to engage in this project where she took a few dozen mice and then did careful brain sectioning at different levels, at different bregma positions, and then used punch biopsies. She went and punched in those little red circles, which are defined anatomical regions for the brain. And then on these little punches, we were able to apply our methods for the mitochondrial health index on 6 different mitochondrial features. So the 4 pieces that I mentioned earlier, plus complex 1, and then the combined mitochondrial health index that makes for 6 features. So this is the basic layout for the brain aficionados who know the mouse brain. These are the bregma coordinates and the different regions. I won't go through the details, but there's a paper on bioRxiv with all of the details if you're interested. So the first thing we wanted to know was, well, are there mouse-to-mouse differences? How large are these mouse-to-mouse differences? And you can see here this little schematic. There are mice that are kind of average, and then there are mice that have low mitochondrial health, high mitochondrial health based on this MHI. So how much difference are there in the mitochondrial content or respiratory train activities? So one way to quantify this here, I'm plotting citrate synthase activity on the y-axis for one brain region, right? Of the 17, this is just one brain region, the substantia nigra. And each dot here is a mouse. So you can see that in this uniform, genetically uniform cohort of animals, there are some mice that have citrate activities of about 600, 500, 600. And then there's one mouse at about 16, 1700, right? And the average is around 800, 900. So you have the mean, 884, the standard deviation, and then you can compute a coefficient of variation, which is the standard deviation divided by the mean, which gives you how much spread there is around the mean. So 29%. And I just want to remind you, all of these animals are syngenic C57 black 6 mouse that all ate the same food. They all grew up in the same environment, breathed the same air, and many of them had the same mom. Yet there is substantial variation in citrate synthase activity in this brain region. If we look at this across all of the brain regions, oops, just going back. This table here illustrates this point, that this 29% in the substantia nigra is not unique. And on average, the coefficient of variation is about 30, 40%. And which is much greater than the technical variation in the assay. Just clicking on the next slide. Okay. So now let's look at the effect of stress. What are the mitochondrial recalibrations to those stressors? And here what you're seeing on the left-hand side is a plot with the effect size. On the left means it's decreased. The complex 1 activity of the mitochondria was decreased with the social defeat. So this is the mice that are bullied for 10 days. If the point is on the right, it means it's increased with social defeat. So this top region, the periaqueductal gray, which is a small brain region located near the middle of the brain, the activity was lower by 42%. So after 10 days of social stress in these animals, the citrate synthase activity goes down by 42% in this one brain region. Then you see what happens in all of the brain regions. So notice that all but one point's brain region is on the left-hand side, right? Which is very unlikely to be due to chance. So on average, social defeat seems like it reduces enzyme activity in the brain. At the bottom there, you have non-brain regions, the liver, the heart, the soleus, the white gastrocnemius 2 muscle, and the adrenal glands. So in the middle here, you have the heat map. If for the 6 mitochondrial features across all of the brain regions, if the cell is red, it means that stress increased the mitochondrial activity. If the cell is blue, it means that stress decreased the mitochondrial activity for that brain region. This is for court, here's for social defeat. Right, so the 2 stressors are inducing different changes in different brain regions, and these mitochondrial responses are very dependent on which brain region we're looking at. Some brain regions, like at the top here, the amygdala, the substantia nigra, experience an increase in mitochondrial activity with the stress, and some brain regions experience a decrease. And that difference is quite substantial between the 2 stressors. So let's go on to the second question then. Does mitochondrial respiratory chain function or content predict animal and perhaps human behavior? So there's recent data that did wonderful experimental work where this top paper here in PNAS from Carmen Sendi's group went in and injected inhibitors or activators of certain mitochondrial processes, specifically in the nucleus accumbens, and examined behavior and specifically social subordination, finding that if you manipulate the mitochondria, you manipulate the behavior of the mice. And then there's this other paper from 2 years ago showing something, a follow-up on this. So let's look at this in our mice. So we have mice with very large variation in mitochondrial content and activity. Does that correlate with their behavior? So what Ayelet did here is to profile the behavior of these mice on 4 different behavioral tests, including the open field test here, for example. Mice that are very nervous and anxious, they will stay in the periphery. They will not often go to the center. And then what this little graph on the bottom left here shows is that some animals have very high anxiety. They never go in the center. And some mice spend a lot of time, a large percent of their time in the middle. So these you would classify as low anxiety. And then in parallel, again, we measured profile of mitochondria, and then we can look at the correlations. Are the differences in behavior related to differences in mitochondria? These are just sample correlations. The first graph here shows complex 2 activity of the mitochondrial electron transport chain. And then on the y-axis is the open field test, how much time is being spent in the center. So what this correlation shows here is that mice with higher mitochondrial activity for complex 2 are more anxious. And you can see the strength of the correlation here. Then the elevated plus maze, which is this kind of plus sign thing that, there's 2 closed arms that feel safe for mice, 2 open arms that feel threatening, and the low anxious mice will go into the open arm. And here we're correlating the mitochondrial health index, specifically in the nucleus accumbens, with this anxiety score from the elevated plus maze. And again, mice with high MHI are more anxious. You can do this not just for these 2 correlations, but across the 17 brain regions, across the 6 mitochondrial features. It gives you these heat map here. So what this heat map shows is that depending on the test, different brain regions are differentially correlated with the behavior. So the null hypothesis here is that this heat map would be white. And if it's random, then there'd be some light blue, some light red, and it'd be kind of very noisy. What you see here is that, for example, in the elevated plus maze, the majority of the correlation, right? More mitochondria means more anxiety. And the frequency distribution of these effect sizes are on the right here. So the elevated plus maze, the open field test show positive correlations for anxiety with the mitochondrial features, whereas the social avoidance test, how socially inclined mice are, show the opposite pattern of correlations. So these data show that mitochondrial content and respiratory chain function in specific brain regions are associated with naturally occurring and stress-sensitive behaviors. Then we can start to look at this not just in a specific brain region way, but to actually bring into this framework the network organization of neural activity that people know very well, especially in the human literature, where there are large-scale regional networks that are made of different brain regions that themselves are made of multiple different cells, which themselves are made of millions of mitochondria making energy and talking to each other. So this is then the kind of matrix that you can generate to look at this connectivity or this network architecture of brain mitochondria, where what you have for each brain region on the left-hand side, so the rows are the 6 mitochondrial features, and then these are just correlated against one another. What you see is that for all of the brain regions, the 17 brain regions, this is generally red, right? So mice that have high mitochondrial activity and content in one brain region tend to have high mitochondrial activity and content in another region. But this is, if all of the mitochondria were the same across the mouse brain, this would be a big block of red. The fact that this is not uniformly red tells us that there are some region-to-region differences. And at the bottom here are non-brain peripheral tissues. And notice that there's a lot of blue here, and there's not predominantly red. So mice with high mitochondrial activity in the brain are not the mice with the high mitochondrial activity in the muscles and in the liver and in the heart. So there's a few questions we can ask here. For example, is mitochondrial activity a property of the animal, or is it independently regulated between brain regions? And like I was saying, the fact that this is all not red shows that there is some variation. There are brain regions that are highly related to one another, like the medial prefrontal cortex, caudal putamen, and the primary motor area, M1. There's also these two, the dorsal dentate gyrus, the ventral dentate gyrus. These are very close to one another in the brain, and maybe as expected, they're fairly highly related. And then there are regions that are negatively correlated. So mice with high mitochondrial activity and content in the amygdala have actually low activity in the vestibular nucleus, which is a brainstem region. And here is another one between peripheral and the brain, where mice with high mitochondrial activity in the hypothalamus have low mitochondrial activity in the adrenal glands. And of course, in the stress pathway, there's important communication between the hypothalamus and the adrenal gland. So some interesting things here that are hypothesis generating. This is the degree of global connectivity. This is a metric that's borrowed from the human neuroimaging literature. The higher the intra-regional correlation means that the more uniform or the more similar to the rest of the brain the mitochondria are. So here we see that mitochondria in the cerebellum have exhibit the highest connectivity across the whole brain. And mitochondria from the brainstem, the vestibular nucleus on the far right, are the least connected. So thinking again about networks, and we wanted to ask, do mitochondria in the mouse brain exist as functional networks? And to do this, we partnered with Manish at Stanford, and he has developed this very cool method that uses here multi-slice and multiplex network analysis. And the actual method that he uses is called topological data analysis, where you can transform this big matrix of numbers into kind of an understandable space of regions and parameters that are connected to one another. So here we can deploy these kind of computational approaches across the 6 mitochondrial features, across the 17 brain regions. And what this leads to in this is this matrix of correlation, which we can visualize in a different way like this. So here, what we're seeing is the association, right? Each node, each little circle is a brain region with all of its mitochondrial biology that was phenotyped. And then the closeness between the nodes, between the brain regions, is proportional to how similar the mitochondria are. And so you can see that, for example, the M1, the primary motor cortex, the orbital frontal cortex, the OFC, the caudal putamen, all the pink circles there at the bottom, they share similar mitochondrial phenotypes, right? And then, so this is one community or one cluster or one network, depending on the term you prefer. Then there's the purple community, the number 2, and then number 3 is a third community, which are more similar to one another. So this is functional grouping of brain regions just based on mitochondrial metrics, right? So this is trying to create some kind of a map of the functional distribution and the connectivity of mitochondria across the brain. We can ask whether this is different than what you would expect by chance. If you just throw a bunch of numbers together and you ask them to create a network, they will create some network, but this one is significantly different than what you would expect by chance. So there's something far from equilibrium. Here, this is the same network, but now the color of the nodes encodes the correlation with behavior, right? So based on the heat map that we saw earlier, now this is projecting this data into this network space. And what this illustrates is that those mito-behavior correlations actually vary by network. And the network or the community number 1 is where the stars are, is uniformly the network where the strongest correlations. So this cluster of brain regions here at the bottom are the regions where the mitochondria are most strongly related to animal behavior. So now, can we create a functional cartography, right, of the mitochondria? The functional data of mitochondria across the mouse brain, the correlation with behavior suggests that mitochondria are not all created equal. Different brain regions have different types of mitochondria, and maybe if we can map this, it would bring us closer to understanding the role of mitochondria in brain and behavior and in psychiatric disorders. So I have one slide on this. This is very much in progress. This is called the Mito Brain Map version 1.0. And the goal is to create a multifunctional mitochondrial atlas of the human brain. And we're starting with a single coronal section. The human brain is just absolutely beautiful. This is a slab post-mortem of human brain, where you see all of the beautiful structures, the corpus callosum, you see the white matter, the gray matter, the cortex, the insula, the putamen, the hippocampus is there. So all of these beautiful anatomy. And now what we tried to do is to examine mitochondrial biology using the mitochondrial health index at fMRI resolution. For those of you familiar with functional resonance magnetic imaging, the resolution of fMRI is 3 millimeters by 3 millimeters by 3 millimeters. So when you slide into the scanner and the scanner starts to scan your brain, it actually creates images at 3 by 3 by 3, so 3 millimeter resolution. So we thought, okay, let's now cut the brain into small cubes of 3 millimeters and then deploy the mitochondrial health index. So that's what we did. Worked with Eugene, my colleague here, who's a chemist and an engineer at heart, and he developed an approach to do this, to actually turn the frozen slab of brain into small little cubes, and then we can do the mitochondrial health index. We can do single nucleus sequence, RNA sequencing to profile cellular composition across the human brain. And then we can work with people who do functional neuroimaging so that we can project this specific brain from this person onto the standard reference Montreal Neurological Institute, the MNI reference atlas that everyone uses in the world. So now we can have a map of mitochondrial biology that can be used by neuroimaging, by the neuroimaging community. So the goal here is to bridge this gap between the subcellular mitochondrial work and the neuroimaging world. And we have an amazing team that did this, the students who went and collected all of the voxels. This is 704 voxels for a single slab of human brain. And then the 5 different biochemical assay for mitochondria. So the dimensionality scales very quickly. And we worked with our colleagues here in psychiatry to have this precious bank of human brains from healthy and psychiatrically healthy individuals. So this is in progress. And the goal, again, is to close the gap between organellar bioenergetics profiling and then whole brain neuroimaging modalities. And I'm just going to say 2 words about this. Now we're also doing this in our MISB study, which is the study I mentioned briefly about patients with rare mitochondrial diseases, where we're doing neuroimaging, structural and functional, to ask how mitochondrial DNA mutations affect the human brain circuitry, both in terms of their structure and function. And this is, preliminary data. We might be able to tell who has mitochondrial disease and who doesn't, just based on patterns of functional connectivity in the brain, which is quite exciting. And now we're working with colleagues who are Alzheimer's disease experts to scale this in the human brain, and hundreds of human brains across multiple regions to bring mitochondrial biology to scale. So in closing, this is the model. This is our mitocentric worldview. We think a lot about how mitochondria produce energy, but also transform and exchange information with one another, with the nucleus, how this in turn informs how cells behave and allows cells, including neurons, to talk to one another. And then this cells talking to one another allow organs to talk to one another and create functional, healthy organisms. And then when you have functional, healthy individuals and organisms, then you can create healthier communities. So this is the vision for how changes at the level of mitochondria can ripple out, right, to the cell, organ, individuals, and community level. And very importantly, I didn't touch on this almost at all today, there is information also coming the other way, right? I mentioned this connection between moods, subjective psychological states, and experiences that actually might materialize into molecular changes inside the mitochondria and changes in energy production capacity. So there's a great need for the young students and trainees that are here today. There's a great need to understand these processes, these psychobiological processes, and the focusing on mitochondria and energy flow has been a productive hypothesis to pursue. And one way in which we think about this is about mitochondria functioning a little bit like the brain. So what neural networks do is they process sensory inputs and environmental signals. Those get integrated and processed by, through neural networks. Then there's process of synaptic plasticity, memory formation, and so on. And the goal of the brain, ultimately, the purpose of integrating information, receiving and integrating, is to produce outputs, intelligent outputs that inform function and the rest of the body, coordinates the organism's functions. So we see mitochondria in a similar way. The mitochondrial network might have the ability to respond and to metabolic and endocrine inputs, to energy levels, respond in changes in shape and function. Mitochondria appear to have the ability to process information, to integrate via their communication with each other in partnership with the nucleus. And some of what I showed you is that mitochondria also can produce outputs that will shape not only cellular behavior, gene expression, the epigenome, but then also stress responses and the coordination of processes in the whole body. So to close, these are the people that have made this work possible. Ayelet did almost all of the work in the mouse study, and she's now a graduate student at the Weizmann Institute in Israel. Our wonderful team of collaborators who know about the mouse brain and helped us achieve this project. And then Eugene, who is kind of the master chef behind the human mitobrain map and the voxelization of the of frozen human brains. And then our colleagues, Phil and Hans, who help us bring this work into the human brain at scale. And Michel Thibault, who's a wonderful computational neuroimaging expert who's working to make the mitobrain map a reality for the neuroimaging community. So thanks very much for your attention, and I'd love to hear your thoughts and questions.
Thank you very much, Dr. Picard. That was fascinating. I'm sure there are many questions percolating in everybody's minds. So we have a question in the chat from Nick Ernst. A really interesting talk. How do you think your social stress results would change if you gave the mice a different stressor, exercise, time-restricted feeding, heat treatment, etc.?
Yeah, this is a great question, Nick. It's, we have two different kind of stressors, but one is social, one is neuroendocrine, right? As a single stress hormone. Those stressors in our models also have very different durations. One is 10 days, the other one was 3 months. We're seeing very profound differences between those two stressors, between the two modalities. So my expectation is that every stressor is going to require a whole brain and a whole body, recalibration that is going to be fairly unique. So the duration of the stressor, the nature of the stressor, might be critical in determining what kind of recalibrations you're going to see in which kind of mitochondria. So that's a good question and something that needs to be explored in more detail.
Thank you. Anybody else? Feel free to unmute yourself and ask the question, put up your hand, and I will enable that. So while we wait for something further from the audience, I'm going to ask you a question. Did you imply that mitochondria from peripheral blood cells would reflect what's going on in the brain? Is that something that I interpreted correctly, or?
So I think it's an assumption that is often made, and it is an assumption that we have made and that we have used as rationale, in previous grants to justify using blood mitochondrial bioenergetics to get insight into what might be happening in the brain. I'm much less convinced now that this is a valid approach for two reasons. One is that mitochondria are so different in terms of their composition between different organs, between different cell types. And we've been doing some work now, building on the well-established differences between cell types. Yeah. In protein and, cell surface receptors and so on. That's for immunologists, for example, it makes no sense to talk about a white blood cell. There's all of these subtypes of B and T cells, and then CD4, CD8, and then some are naive memory and effector. And so there's all this complexity. So all of these cell type differences are critical to our understanding of cell biology. I think we're getting to a point now where we need to start to appreciate that there are also mitochondrial types or mitotypes. And each cell type has different mitotypes. And the blood cells, recently we did some work on phenotyping the mitotypes in different types of immune cells. And the work that I showed in the white blood cells were mixed peripheral blood mononuclear cells, or PBMCs. And now we know better. We know that the mixed PBMCs, the different cell types, have very different mitotypes. And I think it's likely that what we detect in PBMCs is distorted from the reality of what's actually happening in different cell types, unless the same thing is happening in every cell type, which I doubt is the case. So that's the first argument. And the second is these data that I showed in this big, maybe confusing matrix where you have the big red box of all of the brain tissues and then the non-brain tissues at the bottom. The bottom section was not red, meaning it was not positively correlated with the brain. So this is puzzling, but also consistent with some previous data showing that mitochondrial DNA copy number in different human tissues, these studies were done post-mortem, where you have a whole body, you sample different tissues, you measure copy number, and then one person in your cohort of 100 people will have very high copy number in the brain. Then you say, okay, this person as a whole probably has a high copy number. Then you look in the muscle, this person is actually in the lowest decile of the distribution. So copy number, mitochondrial DNA copy number, not well correlated between tissues. And here we've, I think, extended this, looking at mitochondrial content, mitochondrial respiratory chain capacity, and also very poor correlation between the brain and the rest of the body. So this almost suggests that maybe different animals achieve, their state of health through different mechanisms or through different paths. And the combination of mitochondrial content and activities between brain regions, between body tissues, might be different. So this makes me a little hesitant to take leukocyte mitochondrial bioenergetics as directly reflective of the brain.
Right. Okay, we have a great number of questions now. One from Dr. Peter Carlin. Great talk. How tight is the correlation between neuronal activity as measured by fMRI and the mitochondrial signature?
Yeah, this is a long-standing question that we don't have an answer to from our studies, but there's a recent paper about 6 months ago, maybe a year ago in Nature, that did the closest that any group has gotten to this in the Drosophila brain. So they had a little flies and they can image brain activity at the same time as imaging ATP levels, which is kind of a decent surrogate for mitochondrial activity. And they showed very good coupling between the activation of brain circuits, brain and neuronal activity, and then the ATP synthesis that was happening. And the beautiful thing here is that there were some brain regions that were starting to make ATP even before they start to fire, because the other regions were firing. So the brain was anticipating a rise in energy demand and distributing blood flow where it was anticipating that the activity was going to come. So there is coupling, but that's a great challenge, as I think everyone will appreciate, because you need to measure things at very different levels of organization. But great question, Peter.
Great. A question from George Zoidi. What is known about structural or functional difference of mitochondria in pre versus postsynaptic terminals of neurons?
Yeah, this is a great question. I don't know between pre and post. There's very good proteomic work that's been done characterizing the proteomic composition, right? What proteins are in mitochondria and what abundance in presynaptic terminals, where there's a lot of energy required to make neurotransmitters, package them, release them, reuptake them, and then there's a lot of mitochondria in the presynaptic boutons, and then there are mitochondria in the cell body, like those that we saw in 3 dimension, right, around the cell nucleus. So there's been proteomic comparisons of mitochondria between the synaptic and the non-synaptic, which are probably mostly kind of somatic mitochondria, and they're quite different. So there's a nice paper, I think from 2017 or 2014 on this.
And probably respond differently too, right?
Yes, most likely.
Most likely.
Yeah.
Okay, question from Julie St. Pierre. Are there specific electron transport chain complexes that track better with stress?
Yes, good question, Julie. I'm not sure. I'm not sure. We've some of the results that I showed, complex 1, 2, and 4, were sometimes differentially correlated with across brain regions with the different stressors. I think complex 4 was probably the most sensitive to stress exposure, at least where the effect sizes were the strongest. But it's also a fairly robust assay. In the complex 1 activity assay, which is an ADH dehydrogenase. There's a lot of other NADH dehydrogenases in the brain, so the signal for NADH is for complex 1 is not as clean as complex 4. So I'm not sure if the fact that we're seeing stronger responses to complex 4 is because it's a more robust assay, or whether it's something that's biologically real.
Question here from Angela Pachaco. I'm assuming an end goal for some of this research is to have pharmacological therapies for mitochondrial dysfunctions. How does one account for the tissue and region-specific activity and variability in mitochondrial function?
Yeah, fantastic forward-looking question. The tools we have with pharmacotherapy are kind of shotgun, right? You give something, you ingest something, or you inject something, and it goes everywhere, and then you assume that it goes to the right place, or you assume that when it goes to the wrong place, it doesn't do harm. So it's a very challenging question. The organism and the brain has this amazing ability to create functionally, morphologically, molecularly distinct pools of mitochondria that perform different roles in different brain regions. This is kind of next level bioengineering that maybe we'll never be able to achieve, and so I have kind of mixed hopes for pharmacotherapies to have brain region specific or mitotype specific effect, but that could come. I could be proven wrong. So it's challenging, and there might be approaches. There are physiological kind of naturalistic approaches, like not eating too much, or time restricted feeding, or maybe a version of calorie restriction. Physical activity, yes, moving, stimulating your mitochondria, taking the stairs instead of taking the elevator, and walking, and those things are great for mitochondria, not just in muscles that are working, but in mitochondria in the brain. And there's evidence that exercise stimulates mitochondrial biogenesis in different brain regions. So those are probably, and maybe some of our recent work is showing maybe like feeling inspired by life, and positive psychological states might have positive effects on mitochondrial energetics, right? This connection.
What about antioxidants or?
Yeah, antioxidants. I think there's mixed literature there, and there's a compelling pair of papers that show the high dose antioxidants was actually completely blunting the mitochondrial adaptations to exercise training. So if you have a group of individuals train for 8 weeks, and then after 8 weeks, the people that don't take any supplement, they have more mitochondria that function better in their muscle. So you can take a biopsy and measure this. Then you take this is RCT. So the other group, this is a placebo group. The control, the group that received high dose antioxidant have absolutely no change in mitochondrial content and adaptations. So the high dose antioxidant can actually blunt some adaptive processes. And that's probably in part because the reactive oxygen species that are produced from mitochondria are important signaling mediators that are telling the cell, we need to adapt, we need to make more better mitochondria. And then if you blunt this with exogenous high dose compounds, then that can perturb the natural balance, the inner wisdom of the organism.
Okay. Question from Robin. Do you think the stressor-induced calibration has something to do with substrate utilization, unlike other stressors, such as in type 2 diabetes?
Yeah, that's quite likely. So we haven't looked into this. There's a few ways in which this could be done. Different parts of the brain probably have slightly different substrate preferences, but I don't know that anyone has looked at this.
Okay, question from Dr. Andreazza. What are your thoughts on mitochondrial transplants as potential therapeutics?
Yes, I love this. I don't know very much about that. Part of our lab works on cell-free mitochondrial DNA and circulating mitochondria. I think this is an example of a way of intervening. If you were to do mitochondrial transplant, right? You supplement the organism with mitochondria, either from the organism itself or from exogenous source, and then you let the body, you let the brain regions or the different tissues take up those mitochondria as they need. So that seems like a potentially a good way to harness the naturally occurring intelligent processes within the body. And so I've seen some good promising results for this in different contexts. So I'm excited about what's going to emerge from this field in the future.
We have to close the session, but I cannot resist one small question. How is the network established between mitochondria in different regions? Is it like LFPs? Like, what is the connection?
Yes, that's something the reviewers asked us to expand on. We don't know. It could be activity driven, right? Like brain regions that fire together. The classic neuroscience saying is regions that fire together wire together. It could be also that regions that fire together, build and develop bioenergetic properties that align with each other. So that could be part of it. And that model would say that it's brain activity driven, mitochondrial recalibrations and shaping. It could also go the other way around. But at this point, we don't know what could be driving this.
Super. Thank you very much, Dr. Picard. That was very illuminating, and I'm sure generated a lot of good ideas in people's minds. Thank you very much.
My pleasure. Thank you so much.